Mm, I'm gonna do this. I'm gonna do this. And I'm gonna do this. You should know how cells divide. We have two types of division, right? We have mitosis and we have meiosis. We are talking about mitosis predominantly here. We'll talk about meiosis towards the end of the semester when we look at the reproductive system, right? Because mitosis is the one that's happening pretty much everywhere, except with the formation of sex cells, all right? So when we're talking about the cell cycle, we're talking about the phase of interphase, phase of mitosis, followed by cytokinesis. But mitosis is what we're talking about here. And mitosis is division of body cells. Okay? Not sex cells. Sex cells is meiosis, although it's the same stuff for the most part. Okay, but it's just a little bit different. And we'll talk about that when we look at how egg and sperm get made later in the semester here. But here is the cell cycle in all of its fantastical glory. A phase of interphase, which interphase means rest, but in the grand scheme of things, it's getting ready for the division process. As you can kind of see, there's three parts to it: G1, S, G2. I think I have them broken down on the next slide, so I'll just leave that for there. And that's like 90% of a cell's time is spent in that phase of quote unquote rest, prep for the division. Mitosis, okay, is the division: prophase, metaphase, anaphase, and telophase. And those are the kind of four basic phases that we have. Some other textbooks, professors, they'll go into a pro-metaphase. I don't really care. We'll just go with the major four. And then lastly, we have cytokinesis. And cytokinesis is just division of cytoplasm. And some cells do it, some cells don't. Who's got a question?
Sorry. It cut out on me again. You said we didn't need to worry about ubiquitin, right? On protein degradation.
Correct.
Okay, thank you.
Yes, sir.
And I guess while we're pausing, I just wanted to make sure, because I'm still having connection issues. We haven't asked any questions, right?
No, I have not yet.
Okay, just making sure.
When we look at interphase, excuse me. When we look at interphase, it has three parts: G1, S, and G2. And G, in most recent textbooks, refers to gap. You could think of it as a growth phase, to be honest. The cell's getting ready for division. So G1 phase is usually when organelles are starting to double up. Even, the cell's, generally functioning normally, but organelles will start to double up. In the S phase, S means synthesis. And for here, we're talking about synthesis of genetic material. So in this S phase is when we have DNA doubling up. One cell's becoming two cells, right? So everything needs to double up. So in S phase is when we get DNA start to doubling up. G2 phase, anything that didn't get doubled up in G1 doubles up here. Most recent textbooks have discussed it in terms of proteins and whatnot. So whatever proteins that the cell needs for division to occur, you'll see those being doubled up here a lot. So G2 phase, again, you can say growth of organelles that didn't get formed, doubling up of proteins that the cell needs for the division process. That's what you see a lot of occurring here. And there's just a fabulous picture looking at our sister chromatids with our condensed genetic material, right? We have a number of checkpoints. We have a checkpoint in S phase. We have a checkpoint as we enter in from interphase into mitosis. And we have a checkpoint during mitosis process itself. These checkpoints are there to protect defective DNA from being created or doubled up. If DNA is defective, what's the word that we use for that? It's the C word. Cancer. Okay, cancer. A cancer cell is a cell that has lost control of its cell cycle. There's a specific gene, I think it's called P53, but there's a few kind of markers that the cells have to say, okay, you're good. These checkpoints are fantastic because they pause the process of the cell cycle. Okay, they pause interphase from going into completion, or they pause between interphase and going into the phases of division, or they pause the phases of division. If for some reason there is a defect in the read of genetic material, we can stop. We can fix the potential issue. By we, I mean the cell. Can fix the potential issue and start it back up. If for some reason we can't fix it, generally we have a number of different proteins that will bring in to kill it. And that's that programmed cell death, that apoptosis. Okay. Again, as you can kind of see, there's a number of mechanisms for which that happens. Extrinsically, intrinsically, generally it's the intrinsic ones that we're talking about with the cell cycle here. Again, cancer cells, they've lost that control, okay? That gene, P53, is the one that they do a ton of research on. The cell is lacking that ability to pause, to fix, or to kill itself if it can't fix. So it continues to divide and divide, causing that defective DNA to be doubled up in all of the new cells that are forming. Obviously, then that leads us to cancer and all the other nasty stuff that goes with that thing. All right. But let's assume everything's cool and kosher. From interphase, we enter into the first phase of mitosis, and that is prophase. What happens during prophase? That would probably be a good thing to know for an exam. Putting them in the correct order would be a good thing to know for an exam. In prophase, the chromosomes have condensed down or are condensing down. The nucleolus disappears. The nuclear membrane starts to fragment and disappear. We have these things called centrioles, and centrioles do one thing. Centrioles make spindle fibers. There are always two of them. They're always paired. They're always at right angles to each other, and they're always near the nucleus. During prophase, these centrioles start to pull apart from each other. And they go to opposite poles. As they're starting to make that journey, they are secreting spindle fibers. They're producing spindle fibers, just some microtubules. And these spindle fibers go and start to extend toward those centromeres of the chromosomes that are being produced. This process goes through various different adaptations of it. That's where prometaphase is kind of an in-between spot between prophase and metaphase. Okay, if you want to describe it, if I asked it as a short answer question and you wanted to describe prometaphase, I don't care. Knock yourself out. All right. Prometaphase, the chromosomes are continuing the condensing process. Some of them may be at midline, some of them not are at midline, but it's just this in-between phase between the beginnings of prophase and what happens in metaphase. Okay, that's it. In metaphase, these chromosomes are now fully formed. They are fully lined up at the center of the cell. Okay. Interphase up here. How do I know it's interphase? Because I can see that nuclear membrane right around that nucleus. How do I know it's prophase? This is a great picture of prophase. I don't see that line. I don't see that nuclear membrane there anymore. It's fragmented. It's gone. It's disappeared. And now I can start to see some strands, obviously here. Okay, I can see a strand there, but I can't make out. I don't know. I can't really make out anything. But here I can see some individual strands. I can see where it starts, where it stops. Here, these chromosomes are fully condensed down and they're at the middle, the equatorial plane, right? Metaphase plate. Here's a centromere right there. And there's a centromere right there. Okay, a centriole. And there's those little wisp. Okay, those little wispy things right there that are extending from it, emanating from it. Those are the spindle fibers. Okay, those are the spindle fibers. Those spindle fibers have attached to all those centromeres there on the chromosomes and moved them there to the center. In anaphase, chromosomes get pulled apart. And I get one chromatid going to one pole. And I get one chromatid going to the other pole. Okay, so I get one chromatid going this way and one chromatid getting pulled in the other direction.
Dr. Thornley,
Yeah.
do you think, are we going to need to label those pictures of the phases?
Probably not.
Okay, thank you.
Mm-hmm.
And then to go along with that, I had a question.
Yeah.
You said it was matching, and just short answer. It wasn't like fill in the blank, or can that happen as well?
Oh, sure. It could happen. Yeah, that could happen.
Just wondering.
Yeah.
So you said these centrioles are just like little little tubules that just produce spindle fibers.
Yep. That's what it looks like right there. Okay. Always paired, always at right angles to each other, and they only do one thing, and they make microtubules. Microtubule maker. Okay. And those microtubules just start emanating from it. As they start to kind of get produced from these guys, okay, you'll just see kind of these little asteroids extending out from it. As they continue to be made, they go and find, okay, they go and find them right there. You can see it. They go and find and attach to those centromeres. And when those spindle fibers contract, they pull them towards opposite poles. And that's a fantabulous picture of an anaphase. Okay. as these things get pulled apart, this side of the cell kind of goes in that direction, that side of the cell goes in that direction, and they start to pull apart. And you'll see this cleavage furrow right there. There's the cleavage furrow. There's the cleavage furrow right there. And we start to see that in telophase. If you remember prophase, just tell yourself it's the exact opposite. Nucleolus comes back in telophase. Nuclear membrane starts to come back. These chromatids, TIDS, start to uncondense down into chromatin, TIN. And the cell stops its division. Okay. At this point, the only thing that's happening as this thing pulls apart from one another is the division of cytoplasm, right? So cytokinesis starts here during telophase and will continue until the cell divides into two individual cells. Okay.
I'm just making sure. So we're not going to cover anything with G0 or G0.
No. No.
Okay.
Nope. G0 happens for nervous tissue predominantly, where those cells don't divide. These are only for actively dividing cells. And there are only a handful of neurons within the nervous system that are actually undergoing division anyway. So we're not going to spend.
Okay. Thank you.
Yeah, you bet. Again, mitosis is happening in body cells. And with that type of division, guys, we get two identical cells. That is the end result of mitosis. Okay. This is what happens in skin, right? You want skin to become skin. I don't need skin to adapt and become something else. Okay. Liver cells to become liver cells. Muscle cells to become muscle cells. All right. So you get two identical cells. Now in meiosis, we get adaptation. We get variation. Okay. We get 4 haploid daughter cells, and we'll talk about meiosis later in the semester. Okay. But that is the end result of mitosis. Two identical cells. Yeah, there's a centrosome. Again, this is 2 centrioles together. Gosh, I cannot write today. Okay. And it just makes spin files. And that's what it looks like. Okay. Cells can only divide for so long, and then they stop. We have these things on the end of on the end of the DNA molecules called, where's that? Called telomeres. And it's those telomeres that help to maintain the integrity of the DNA molecule. If telomeres are lost, which they are every time a cell divides, that increases the wear and tear on a cell. And it's what helps to drive basically apoptosis as well. Okay. If I can maintain the viability of that telomere, then the cell will divide forever. Again, cancer cell, the cell is immortal. Okay. So the cell will continue to divide and divide and divide. Genetic material be damned. It's already defective. Okay. But these telomeres are what actually help to govern the length of time that these cells can divide. Okay. And again, as you get older, right, cells wear and tear and break down, and it's the result of these telomeres that help to govern that aging process that occurs normally with cells. Can you take creams? Can you eat better to prevent excessive signs of aging and wearing and tearing on you? Yes, you can. Okay. So there is that aspect of life. There's gene therapies and whatnot, I believe, as well, that are involved in this process. But for the most part, everything has a normal shelf life for how long they can divide, and it's those telomeres that help to govern that aspect. These are just some terms, guys. Hypertrophy is an increase in cell size. Hyperplasia is an increase in number of cells. Okay. I love to ask things where they're very similar, but like one word difference. Number of and size of would be this just subtle difference between these things. Terms get asked a lot on various different exams of mine when they're one word difference. So just a heads up if you've never had me before. I do like to ask that type of stuff. Oh, here's what the chromosomes look like. If you haven't seen this before, here's a karyotype of what some of the chromosomes look like here. Okay. Where mitosis was involving body cells, again, meiosis only occurs, there it is, in the sex cells, okay, in the ovaries and testes. It's the only place that it occurs, okay? So meiosis is only involved with the making of an egg and a sperm. There are two divisions, okay, a meiosis 1, a meiosis 2. I will beat you to the punch, okay? I never ask a short answer question based on telling me about meiosis. Okay. I never do. And I'm not just saying that and then pull the old rug out from underneath you. Okay. I won't ask it as a short answer question. Could I ask it as a multiple choice question? Yes. What would I ask? The division of meiosis is involved with the formation of what? Egg and sperm. Meiosis occurs where? Only in the gonads, only in the sex cells. Okay, only in the ovary and the testes. Okay. Mitosis has one set of division. Prophase, metaphase, anaphase, telophase. Meiosis has two different divisions. Same phases. We just put the moniker 1 or 2 with it. The great thing about meiosis is that it provides variation. It is not an exact copy like we get in mitosis. Again, skin becomes skin. I don't need skin to become something else. Liver cells become liver cells. Kidney cells become a kidney cell. All right. I don't need variation for anything like that. In meiosis, I get variation. How do I get that variation? I get that variation by a couple of things, but the one I have a picture of, I believe on the next slide, is what's called crossing over. When I cross over, there you go. Okay. As you can see, the different colors there are swapping from one chromosome to another. That gives me a totally different read. That gives me variation. And I can't even remember the technical amount of crossing over and independent assortment, what those two things provide. It's like some kind of 40 octillion different variations of the genome. It's a lot. Okay. It's a number that I didn't even know how to pronounce. I had to put it in Google to give me all the derivation of all the decimal spots and places. So this gives me variation, okay, which is great. Those variations give adaptations, and those adaptations could be advantageous, could do absolutely nothing. But it's what gives diversity to the genome, which really hasn't changed in several million years. There's the steps. I'm not going to ask. I mean, it's still the basic things. Prophase 1 or prophase 2, it doesn't matter. Nuclear membrane fragments, nucleolus disappears, yawn. In metaphase 1 or metaphase 2, the chromosomes are still lining up in the center. They'll have slightly different names, but that's not the goal of this class. You can see it's called a tetrad up there in the first and the second phases of division there. I don't have a tetrad anymore. But I'm not going to ask you that question, guys. We have some complex stuff coming up in the next unit, in the next lecture period, that I can ask more short answer questions on. Okay, so I don't ask meiosis as a short answer question. In anaphase 1 or anaphase 2, they get, the chromosomes get pulled apart. Well, in here, the tetrad gets pulled apart in metaphase 1. I'm sorry, in meiosis 1. And down there in anaphase 2, it's the chromosomes that are getting pulled apart. The end result of meiosis are 4, I'm going to throw in a fancy word in here, haploid daughter cells. Okay? Haploid means they have half the number of chromosomes. Half from mom, half from dad, gets you. And that gives you the 23 pairs of chromosomes that you need. Okay? So the end result of meiosis is genetic variability and 4 haploid daughter cells. 4 sperms. Same thing happens for egg, but for you women, it's a little bit more complex. but you still get 4 ova made. Only one goes to fruition and gets ovulated. Okay. The rest become polar bodies, but we'll get into all that fun sex stuff later in the semester. Okay. Mitosis, body cells. Meiosis, sex cells. Mitosis, identical. Meiosis, not identical. Mitosis, 2 individual cells are made. Meiosis, we get 4 cells made. I like to ask the phases of mitosis. I like to have you put them in the correct order. I like for you to tell me a little bit about each little step and what's happening in each one. Okay. When it comes to meiosis, I like to know how many divisions, 2. I like to know where it's happening and what you get out of it. So that makes a great matching question. I guess it also makes a good fill in the blank question as well, if you want to split hairs. Okay. But I usually ask it as multiple choice.